Good morning. I'd like to thank the Alliance for Regenerative Medicine for giving me this opportunity to share the update with VentriNova today. So VentriNova is a startup company aimed at developing clinical treatments for cardiac regeneration. And this is based on my research program, which first began at Columbia University and spanned a decade at Columbia and now at Mount Sinai School of Medicine. In terms of what the company is, it's a preclinical stage, I should say late preclinical stage, regenerative medicine company that's focused on reversing cardiac damage by activation of the intrinsic repair pathway via cyclin A2 to generate de novo heart muscle cells. I want to say up front, this is not a stem cell-based company, but it is a regenerative medicine company. So cyclin A2 is a key switch that mediates cardiac cell division and tissue growth during neonatal development. And the gene is basically silenced right after birth in all mammalian species, and therefore unavailable or lies dormant and cannot promote cellular regeneration and tissue repair following injury to the heart, such as a myocardial infarction. VentriNova's therapeutic strategy is centered around the development of gene therapy as a first-generation product to reactivate cyclin A2 gene, the native cyclin A2 gene, and promote tissue repair following a heart attack. Preclinical work has demonstrated that modulation of cyclin A2 stimulates endogenous growth of new myocytes and significantly enhances cardiac contractile function in both small and large animals. So as the company was founded by myself in 2006, the therapeutic pipeline is based on the role of cyclin A2 in mediating cardiac mitosis. And this is a key developmental switch, and the early role in cardiac tissue development was discovered by myself, first starting with my postdoctoral time at Columbia University, mentored by Dr. Deborah Wolgemuth, and then later as Florence Irving Assistant Professor at Columbia University. I'm currently Associate Professor at Mount Sinai, and I direct their Cardiac Regenerative Medicine program. So I'm a full-time cardiologist. I also see patients every week, and therefore I understand both the scientific and the clinical spectrum of cardiac regeneration. The company has 3 issued patents so far and several PCT applications in prosecution. Our legal representation is via Wilson Sonsini, Goodrich, and Rosati. And funding thus far, we obtained an STTR grant in 2009 for $500,000. It was the highest score seen nationally over a 5-year period by the National Heart, Lung, and Blood Institute. And we have seed investment from Broadview Ventures. This funding, though, so far has been used to demonstrate the efficacy of cyclin A2-mediated cardiac repair in a porcine myocardial infarction model, along with characterizing the cellular mechanisms of the repair process. I'm proud to say that VentriNova was selected as one of the 50 most innovative healthcare companies globally by TEDMED 2013, and I had the opportunity to present this at TEDMED earlier this year. The company now seeks a Series A round, basically to get us to IND and initiating a Phase 1 clinical safety trial. So the team thus far, as I mentioned, I'm myself am the founder. I have a scientific advisory board comprised by Deborah Wolgemuth, who is a professor of genetics and development at Columbia University. She's also my research mentor from my postdoc days and my assistant professorship. Her laboratory discovered cyclin A1, which is a germ cell restricted cyclin A, so she has a very extensive understanding of the functions of the A-type cyclins. Judy Swain was my mentor during my cardiology fellowship at the University of Pennsylvania. At the time, she was chief of cardiology at University of Pennsylvania, later chairman of medicine at Stanford University, and then dean of translational sciences at UC San Diego. She is currently the executive director of the Singapore Institute for Clinical Sciences. Roxana Meron is a professor of medicine at Mount Sinai School of Medicine. She's also an interventional cardiologist who's very renowned for her clinical trials research in interventional cardiology, and she works very closely with the FDA to develop standard definitions for clinical trials in cardiovascular disease. In terms of the Management Advisory Board, this is comprised of Farhan Sharaf, who is Senior Managing Director of Guggenheim Partners in New York City. He has extensive knowledge of the equity markets, and has conducted analysis of multiple companies, sectors, and markets. I'm also happy to say we've gotten quite a bit of guidance from Rob Margolin, who all of you know from the Alliance for Regenerative Medicine. He has a very extensive understanding of the regenerative medicine business sector, so it's been really fun working with him. So in terms of myocardial infarction and heart failure, as you know, this is a significant unmet medical need, and as you all know, it's the #1 killer in the industrialized world still. Approximately every 25 seconds, an American will have a myocardial infarction, and every minute, an American will die of one. And this is just in the United States. As you know, this is, global health burden. Annually, there are an estimated 785,000 new and 470,000 recurrent myocardial infarctions in the United States alone. The mortality morbidity costs in the U.S. alone aggregate to approximately $177 billion. And the total inpatient hospital cost for cardiovascular disease is approximated at $71.2 billion, which is approximately a quarter of the total cost of hospital care in the United States. Medical costs for healthcare services, medication, lost productivity of those afflicted with coronary disease is in the range of $316 billion, and these statistics are from the American Heart Association and are projected to only keep growing. So in terms of our first lead candidate, VN100, this is a gene therapy candidate, and this is a viral vector-based gene therapy that induces cardiomyocyte division in adult heart tissue by delivery of the cyclin A2 gene. As I said, this is the principal gene regulating cardiomyocyte mitosis. So first of all, we have targeted adenoviral delivery of human cyclin A2 to cardiomyocytes. We deliver this with intracoronary injection to the infarcted area, and it's been tested both in small and large animals. And I'd like you to play the movie. This is an adult porcine cardiomyocyte, and the red is a fluorophore that is tagging alpha-sarcomeric actin, the key structural protein in the cardiomyocyte muscle unit. And that's a cardiomyocyte undergoing mitosis as captured by time-lapse imaging. Now, I'm just reminding you, this is an adult differentiated cardiomyocyte. For centuries, scientists thought that this kind of cytokinesis of an adult differentiated cardiomyocyte was not possible. Well, we've cultured the adult porcine myocytes in a petri dish, treated them with the same viruses that we are giving in vivo, and showing that they do undergo cytokinesis. And this data basically is in a manuscript that is under invited revision at Science Translational Medicine. We're anticipating it will be in press any day now because the revisions are back with the editors. And in this, we have shown that not only do we have cytokinesis of the adult cardiomyocytes, we have preservation of the sarcomere structure. If I can show you right here, that you can actually see the individual sarcomeric units. This is before the cell divided. This is basically a lower intensity picture of the cell before it's divided. After it has divided, the cells were fixed, and you can see the sarcomere is even present in the daughter cells after the division. And this is basically just to show that the nuclei of those individual cells, the nucleus is here for this one, and we've just focused on a region to show the sarcomere structure. We've also confirmed expression of troponin in these cells, just to show another cardiac muscle marker in the cells that we have been studying. And so there we've captured the precise mechanism of how this gene therapy is working. Now, in terms of the in vivo data, we've used every imaging modality at our disposal to show an dramatically increased cardiac function in the pigs that were treated with cyclin A2 compared with the control pigs, which were treated with the same viral vector. It was just missing the cyclin A2 gene. So we call that the null virus. It has the same backbone, but it's just missing the cyclin A2 cDNA. Oops, if I go back. So we've used LV angiography, 2-dimensional echo, 3-dimensional echo, and MRI. If you focus on the last panel here circled in the red, by MRI, the EF decreases in the null-treated controls and goes up close to 20% in the cyclin A2 treated animals. Like I said, these are part of an upcoming manuscript in Science Translational Medicine. And in terms of in vivo cellular histology, we've shown that there's a significant amount of mitosis. Here we have tissue staining with alpha-sarcomeric actin in tissues from the treated pigs. The blue is a nuclear signal DAPI, and we've used the anti-phosphohistone H3 marker, which is specific for cells in mitosis. This is a merge of all three panels, and we get a significant increase in mitosis in the cyclin A2 treated animals. So therefore, we now have 12 years of science supporting a critical role for cyclin A2 in 3 distinct animal models. My very first publication as a postdoc was basically showing that in a transgenic mouse model, which had constitutive cyclin A2 expression, that when these mice were subjected to a myocardial infarction, there was significant repair by MRI. We showed significant increases in ejection fraction. We were also able to show in the mouse model that cyclin A2 was expanding the pool of committed cardiac progenitors. And therefore, you have more progenitor cells that were adopting the cardiac lineage were undergoing more rounds of cell division. So now we think there may be 2 operative mechanisms. The cyclin A2 is inducing cytokinesis in the adult cardiac myocytes, and any endogenous progenitors that are homing to the infarct zone, as they differentiate along the cardiac lineage, they have an enhanced proliferative stimulus, and therefore we get repopulation of cardiomyocytes. We've proven the ex vivo delivery in a rat infarction model, and now I just showed you the results from the porcine infarct model. It's not, thank you. If you could please play the movie. And this is basically an animation produced by Biodigital Systems in New York, demonstrating how our first generation product would be administered, likely through the coronaries in the cardiac cath lab. The black area in the heart is the infarct zone, and you can see in the green liquid denotes the gene therapy being administered down the coronary. The movie stopped, sir. And then it takes you into a close-up inside the artery, where the golf balls basically are the carriers of the gene therapy, with the green particles being emitted from those carrier. And this is with cells that are homing, endogenous cells that may be homing to the area, and the surrounding healthy adult cardiomyocytes undergo cell division and repopulate the infarct zone. Can you hit play again? And that heals the infarct zone, and these are the kind of results we've seen in all animal models that we've tested thus far. So in terms of an intellectual property summary, as I mentioned, we have 3 patents that are awarded already about the use of cyclin A2 in cardiac repair. We have also covered, we also have a patent that's been awarded covering endogenous side population cells in the heart that are responsive to cyclin A2, and we have a number of pending patents in multiple markets around the world. In terms of the VN100 product development overview, milestones we've highlighted over here, which VN100 vector development, large animal trial, characterizing the cellular mechanisms of repair. These have all been done, and now we are constructing a GLP-compliant vector for the large animal. So all of this has been done with the funding that we've obtained thus far. We are now seeking a round A for an IND submission, and we will also be publishing very soon molecular silencing mechanisms of the endogenous cyclin A2 gene, which is the basis for VN200, our second generation product. And that's basically regulating the endogenous gene itself in a targeted manner, not via delivery of a gene therapy product. Since I'm running short of time, I'm not going to talk about rounds B and C. And basically, we've thought a lot about a phase 1/2 safety evaluation. Like I said, this is where Roxana Meron's guidance is critical as she designs clinical trials in cardiovascular disease for the FDA, and we've estimated the costs for conducting a phase 1 trial. And we're basically aiming to include 25 to 50 patients to test safety and dosage. So I'm just going to touch about the VN200. We've basically delineated the epigenetic mechanisms that silence the endogenous gene, and we know that it's a tissue-specific silencing process because cyclin A2, the gene responsible for mitosis, is not silenced in your skin or your intestines or your liver or other organs where cell division is happening, only in the heart, and we know exactly what silences it in the heart. This will be an RNA-based therapy, and we're going to be publishing a paper about that later in this year. The regenerative medicine competitive landscape, I think many of you may know something about these companies, and some of them are presenting here today. I just want to emphasize again that this is not a cell therapy-based company. If you follow the news for clinical trials of cell therapy, you're probably well aware that they have mixed results, and I think even the strongest proponents of cell therapy for heart disease will tell you that these cell types that have been tested in clinical trials are not actually differentiating into cardiomyocytes, nor do they really show significant cardiac cell division of the endogenous cardiomyocytes themselves. So I think we're the only company that does that, and that's why we really stand out in the cardiac regenerative medicine field. And here is basically just a summary in terms of why I think this company is going to succeed. We've demonstrated significant cardiomyocyte division with significant ejection fraction recovery in large animals. This is unparalleled by cell therapies and any other regenerative medicine technologies. We have 3 distinct therapeutic approaches to regenerate heart tissue. We have regulatory routes that exist in Europe and the United States. For our VN100, the first gene therapy product has been approved in Europe, and gene therapy is now gaining a stronger foothold in the United States as well. There's scalable manufacturing. The technology is incredibly novel with a broad IP portfolio, and there are proven scientific and management teams on board. Basically, the growth and exit strategy would likely be a trade acquisition by a cardiovascular-focused large pharmaceutical company or partnering with a larger specialty biotech pharmaceutical firm for joint development. And I think we're in discussions right now with a number of pharmaceutical companies as well for potential partnership. Basically, in terms of regenerative medicine, I think this group is a believer. I don't have to convince you. The sector delivered 3 $1 billion thresholds in 2012, and I think this is a really exciting time to be in the regenerative medicine space. And I know I'm being asked to quickly finish, so I'll go on. And we've just highlighted similar exits within the last few years in the cardiac regenerative space here. Yes. Okay. And I basically just want to acknowledge, just get to my acknowledgement slide. My laboratory, the members that have contributed to this work are mentioned here, my scientific collaborators, in particular my mentor from my research postdoc, Deborah Wogemuth, and as I said, funding is provided by Broadview Ventures and the NIH. Thank you very much.